Now you go to the Chad-Ox result, which is a chimpanzee um, adenovirus. And here you do see not only a lot of immunity, but you also do see T cells. But when they try to boost, all of a sudden you see no effect of the boost. And that speaks in my mind to the fact that it is a xenograph chimpanzee adenovirus. And the moment you inject yourself once with it, you're going to have immunity to the adenovirus. The other disturbing thing with that paper was you begin to see in the elderly very little immunity, both in China as well as in the Oxford trials. Very little, 9% immunity.
Can I ask you a question, though? I was looking at the Ox because that's the one I've seen the most of because the Lancet paper was published yesterday, so I get to look at all of it.
Yeah.
I thought the oldest people in those trials were like 55 or something. Were there anybody older in those?
Even the Chinese paper was published yesterday as well.
Oh, I didn't see that. Okay, maybe I missed that one.
That they did this. They differentiated. Maybe not yesterday. It was, I don't know when it was published. Published recently,
right?
By age, and this was disturbing because greater than 55 is where the risks are.
Right.
But just focusing on T cells. You don't see the T cells, you don't see the, and what's also very difficult, which is unfortunate, it's hard to compare paper to paper to paper to paper, because each of them have different methodologies of addressing their what they call arbitrary units, apples to apples, orange to orange, in terms of the scale of the immunity, etc.
Well, and to your point, actually, about the China study published in the Lancet, it's those 85 neutralizing antibodies, there's that pre-existing immunity there as well. And it was 52% had high pre-existing immunity of the 508 participants, and then 242 had low pre-existing immunity to the vector. And so is that something that could hamper the specific immune responses to vaccination if it's not adjusted for, particularly the humoral immune response?
Well, when you looked at that data, frankly, you just look at head-to-head, there's a significant drop-off based on the ad immunity. And then, if you look at the ad immunity around the world, the US luckily is about 30%, Africa could be 80%, the rest of the world is between 50 to 60%. So if you have ad immunity, this is really a problem if you can use what I call the first generation ad5. So I see, the word is not problems. I see if you had, at least from my view of the science, the ideal world of generating what I consider the ideal vaccine would be a vaccine that, one, could generate humoral immunity, but largely very potent neutralizing antibodies. Because if you look at the neutralizing antibody versus the total antibody level in the Chinese paper, even it's 40 to 50% is only neutralizing antibodies. So that was another thing you need to look at. So antibodies that are neutralizing, but antibodies that are not just IgG, but IgM and IgA. So how would you create mucosal antibodies? Because this is a mucosal disease. It's a respiratory disease. So when you inject something subcutaneously or intramuscularly, you're not going to get mucosal immunity. You need to give that orally. So we're back to something which I'm going to take you down. And the third one is, we know what the epitopes are with regard to the S and the N. And it turns out that the most T cell epitopes are both the S, N, and M. But S mutates, but N doesn't. So N is highly conserved.
There's NSP7 as well. There's NSP7 too, Pat, if I'm not mistaken.
Correct. So the non-structural proteins.
Right.
So we have to have a figure. If I had my druthers, I'd have, and this is where we're going. So it's a little self-serving because that's exactly where we're developing it. But we're developing it based on what I consider, in my mind, the ideal science. To have an adenovirus vector that can be given multiple occasions in the face of adenoimmunity, that has been proven to do so. And this is where Adam and I in the field of cancer have done that. So we've taken this adenovirus, which we have got what we call 4 deletions. So it makes no fibers. So when you inject it into the body, it actually goes to what we call the CAR receptors, the coxsackie adenovirus receptors, and goes in and then sits there stealthily and actually produces its gene construct. We've done this in 125 cancer patients, and we've given them CA MUC1 brachyury, which are proteins, if you put them together, could almost be a universal vaccine for cancer, because most cancers express one of these, including PSA. And we were able to show, when giving them every 8 weeks, think about that, over a year, we were able to generate specific CD4 and CD8 T cells to these self-antigens. So we even broke tolerance in those.
Break tolerance, that was the answer.
Yeah. So
Break tolerance.